Hello and welcome to our video on cellular metabolism, and we're going to be covering four major topics: the metabolic basics, Gibbs free energy, ATP, and enzymes. So start off with the basics, obviously. So metabolism is just the sum total of all the chemical reactions that occur in an organism. They can be catabolic or anabolic. So catabolic means that you take a larger molecule such as glucose and you break that down into its constituent parts. Whereas anabolic would be if you're trying to take the small parts and build them up into a larger part, like glucose. For example, in photosynthesis, you would be that would be anabolic because you're trying to build up the glucose. Whereas cellular respiration would be catabolic because you're taking the glucose and you're trying to break it back down into its parts so you can generate energy. Now, what is energy? Energy can be defined as the capacity to cause change, because when you release energy, it changes the environment around it in some way or another, so we can call energy the capacity to cause change. So let's apply a few principles from physics to better understand natural phenomenon. So we can start with the first law of thermodynamics, which says that energy cannot be created or destroyed. Now, this is pretty obvious because all the energy that's been around since the Big Bang will always be around. The only thing we can do to that energy is change it from form to form. So we can transform it, but we cannot create it or destroy it. Now, the second law of thermodynamics is that spontaneous reactions increase the entropy of the universe. Now, this might sound really confusing, but a spontaneous reaction is one that can happen without the input of energy. So, for example, let's say we have a glucose molecule, and we want to break that down. Now, that can happen without the input of energy because it doesn't require any energy to go from a glucose molecule to the smaller parts. Whereas if you're trying to build a glucose molecule, then that would require energy. Now, these types of reactions that don't require energy will increase the entropy of the universe. In other words, they're going to make the universe more disorderly. They're going to release energy, and that sometimes that energy can be used to destroy things, for example. So that's why it increases the entropy of the universe. Now let's move on to Gibbs free energy. So a living system's free energy is just energy that can do work in cellular conditions. Now, not all energy can do work. For example, organisms often release heat as a consequence of a chemical reaction. Now, that energy doesn't count as free energy. It still exists, but it's not free energy because it can't do work. Now, here's a formula that may be useful. So this is delta G. Now, delta, as you may know, it means change. So, new minus old. So delta G equals delta H minus T times delta S. So G is the Gibbs free energy, H is the enthalpy, which is the total internal energy, T is temperature, and S is entropy. So you can plug in the values here to get delta G, and if delta G is negative, that means the reaction is exergonic, which means that it doesn't require the input of energy. Therefore, it is spontaneous if it is so exergonic. Now, if G is positive, that means the reaction is endergonic, which means that it will require the input of energy in order to be executed. Now, you may be wondering, where are we going to get this energy to power an endergonic reaction? Well, one way we can get that is through ATP. Now, ATP stands for adenosine triphosphate, and so as it's often called the energy currency of the cell. And this is not necessarily because ATP is some magic molecule. It's because its chemical bonds have potential energy. It's not the molecule itself, it's the bonds. And if the bonds are broken down, that's what releases lots of energy, not the molecule itself. That really needs to be emphasized because that's a common misconception lots of people have. Now, the way to break this down is through hydrolysis. So hydrolysis uses water to break the bond, and this results in ADP, adenosine diphosphate, which has two phosphate parts. And then we have PI here, which stands for inorganic phosphate. This is a common notation used to note an inorganic phosphate, which means that it's no longer connected to a carbon. So now let's talk about enzymes. So activation energy is simply the energy that's needed to break the chemical bonds. So every reaction has a different activation energy, and once that energy is provided, then the chemical reaction can proceed. But however, this has to be supplied from the outside. Now, but obviously if you think about it, in an organism, some of these reactions have really high activation energies. So it's going to be really impractical for an organism to attain these massive amounts of energy because that's simply not possible. That's why many organisms, in fact all organisms, have enzymes. And enzymes, what they do is they simply lower the activation energy needed for a chemical reaction. So they bind at an active site, which is where they're used to a substrate. Substrate is what they're binding to. And so they change the shape of that substrate so that those bonds are strained so that the chemical reaction can proceed or get started with less energy than would normally be required. Now, obviously, it would be bad to have all the enzymes active at once because that can cause many devastating effects. So therefore, enzymes often have inhibitors, which block the functions of the enzymes. Now, competitive inhibitors, what they will do is they will bind to the active site directly, and so they're going to be competing with space with the enzyme on the substrate. Whereas non-competitive inhibitors will bind at another place on the substrate, and what this does is it changes the shape of the active site so that, first of all, it'll make it hard for the enzyme to even bind on there, but even if it does bind, it'll make the enzyme work less efficiently. So there's another type of regulation. It's called allosteric regulation. So this is where regulatory molecules bind to the enzyme, and this affects their shape and their function in various ways, which will be discussed later in later videos. So that's all for our video on cellular metabolism, where we covered metabolic basics, Gibbs free energy, ATP, and enzymes. Hope you like our video. Please subscribe and like.